Imagine having a molecular pair of scissors that can cut DNA with the precision of a surgeon's scalpel, but at the nanoscale level. That's exactly what we're dealing with when we talk about CRISPR-Cas systems. These remarkable molecular machines didn't start in a laboratory. They evolved over millions of years as an adaptive immune system in bacteria and archaea. Think of it as nature's own antiviral software, where prokaryotes developed this sophisticated defense mechanism to remember and destroy invading genetic material. The beauty lies in its three-stage process. First, the system captures pieces of foreign DNA, like taking a mugshot of an intruder. Then it processes this information into a usable format. And finally, it seeks and destroys any matching genetic sequences that dare to invade again. Among all the different types discovered, the Type 2 system featuring Cas9 has become our molecular workhorse for genome editing. Now let's dive into the architectural blueprint of this system. Picture a genetic filing cabinet where each folder represents a memory of past invasions. That's essentially what a CRISPR array looks like. These arrays are fascinating structures where short DNA sequences, called spacers, are neatly organized between identical palindromic repeats, much like bookmarks separating different chapters in a book. Each spacer is like a wanted poster, containing the genetic fingerprint of a previous viral invader. What makes this system truly elegant is how it's organized. There's a leader sequence at the beginning that acts like a genetic on switch, containing all the regulatory elements needed to kickstart the transcription process. Those palindromic repeats aren't just spacers. They fold into hairpin structures that are absolutely crucial for the system to recognize and process the information correctly. The guide RNA is where the magic of specificity really happens. Think of it as a molecular GPS system that navigates Cas9 to its precise destination in the vast landscape of the genome. We've engineered something called a single guide RNA that's essentially a fusion of two natural components. It's like combining a map with a compass into one sophisticated navigation tool. The critical part is that 20 nucleotide spacer sequence. This is your postal code in the genome, determining exactly where the molecular scissors will make their cut. But here's the catch. Even with perfect address matching, Cas9 won't cut unless it sees the right license plate nearby. That license plate is called a PAM sequence, protospacer adjacent motif. And for the most common Cas9 variant, it's looking for an NGG sequence right next to the target. No PAM, no cut. It's nature's safety mechanism. Let's examine the molecular architecture of our star player, Cas9. This protein is essentially a sophisticated molecular machine with specialized departments, each with its own job. Picture it as having two separate cutting units. The HNH domain acts like one blade of the scissors, specifically targeting one strand of the DNA double helix, while the RUV-C domain serves as the other blade, cutting the opposite strand. But Cas9 isn't just about cutting. It has this recognition lobe that's like a molecular hand, carefully grasping both the guide RNA and the target DNA to ensure everything is properly aligned. Perhaps most fascinating is the bridge helix, which acts like a molecular sensor, undergoing dramatic shape changes when the correct target is found. It's like a lock mechanism that only opens when the right key is inserted. The actual cutting process is a choreographed molecular dance that's both elegant and precise. It all begins when Cas9 encounters that essential PAM sequence. This is like finding the right keyhole that allows the system to start unwinding the DNA double helix. As the DNA unwinds, it forms what we call an R-loop structure, where the guide RNA can slip in and test for complementarity with the target sequence. It's like trying on a glove. If it fits perfectly, the system gets the green light to proceed. Once that perfect match is confirmed, both nuclease domains spring into action simultaneously, creating a clean, blunt-ended double-strand break exactly 3 base pairs upstream from the PAM. This precision is remarkable. We're talking about cutting a molecule that's only 2 nanometers wide with surgical accuracy. But cutting DNA is only half the story. What happens next determines whether we achieve our desired genetic modification. The cell immediately recognizes this break as damage and rushes to repair it. But here's where we can influence the outcome. The default repair pathway is called non-homologous end joining, which is fast but somewhat sloppy. It often introduces small insertions or deletions that can disrupt gene function. However, if we provide a template DNA sequence, the cell can use a more precise repair mechanism called homology-directed repair, allowing us to make exact changes, like correcting a disease-causing mutation. There's also a third pathway called microhomology-mediated break-induced replication that can create more complex modifications. The fascinating thing is that the cell's choice of repair pathway depends on where it is in its life cycle and what molecular tools are available at that moment. While Cas9 has been our molecular workhorse, the CRISPR toolbox has expanded dramatically with variants that offer unique advantages for different applications. SB-Cas9 remains our most popular choice. It's like the reliable sedan of genome editing, well-characterized and versatile, with its NGG PAM requirement. But sometimes we need something smaller for delivery challenges, which is where SA-Cas9 comes in. It's more compact and recognizes a different PAM sequence, opening up new targeting possibilities. Then there's Cas12a, formerly known as CPF1, which is like upgrading to a more sophisticated cutting tool. It prefers T-rich PAM sequences and creates staggered cuts rather than blunt ends, potentially offering enhanced specificity. And pushing the boundaries even further, we have Cas13, which breaks the DNA-only paradigm by targeting RNA instead, opening up entirely new possibilities for transcriptome editing and regulation.